I'd like to welcome everybody today. I'm Matt Coleman. I'm a senior scientist in the Biology and Technology Division here at Lawrence Livermore National Labs. I'd like to thank you for joining today's LLESA Author Series virtual event. The event is being recorded, and we ask that you type all of your questions into the Q&A field over on the side of your screen. We're also holding a drawing for a chance to win one of Chris Colby's books. And I'm excited to introduce him today because I've known Chris for well over 25 years as a graduate student, as a scientist, and as a brewer and editor, and of course, friend. Chris earned a PhD from Boston University in 1997. His thesis work involved studying intra-array variant movements in the histone gene family of Drosophila and the population genetics of so-called adaptive mutations. His undergraduate degree is in biology and chemistry. So he's actually a well-established scientist, but he's also a brewer. For the past 20 years, he's been a writer and editor for publications dealing with beer brewing and especially home brewing. He's written 2 books on brewing beer, Brew Recipe Bible, and Methods of Modern Homebrewing. And he recently put out a book on making hard seltzer, and that book was titled How to Make Hard Seltzer. He's also written extensively about gardening and raising monarch butterflies. And he's currently working on a book that takes advantage of both his science and brewing background titled Science in the Craft of Brewing, which will hopefully be released within the next year. And he lives with his wife and their many cats in Bastrop, Texas. I'd like to now hand it over and have everybody welcome Chris to our forum. All yours, Chris.
Lawrence Livermore National Labs for inviting me, and especially Matt Coleman for giving them the idea that they could invite me. So I want to talk about the basics of modern brewing. The talk could probably also be titled the basics of modern brewing in the Western world, because there's some differences, but we'll just go with basics of modern brewing. I'd like to start out with a very short overview of the whole process, and then back up again and step through it and give more details. I don't have a PowerPoint presentation associated with this, but I do have the handout file that Colin mentioned. That gives everything I'm going to say in this talk is there, plus about material for like an extra hour of me blathering on. So before we get into the brewing, it's probably a good thing to define what beer is, just to make sure we're all on the same page. Okay, beer is a beverage made from fermented grains. Okay, this contrasts with wine, which is made from fermenting fruits, and most often that's grapes. Mead, which is fermented honey. There's ciders, which is fermented apples. Apples are a fruit, but somehow ciders get their own group. I guess wine isn't a monophyletic group. And there's also sake, which is fermented rice, which ironically gets called rice wine, even though rice is a cereal grain. So the very basics of brewing, you can think of beer as being brewed from 4 ingredients and having 4 primary steps. The 4 ingredients are grains, hops, yeast, and water, and the 4 steps are malting, mashing, boiling, and fermenting. Okay, the most common grain by far used in brewing is malted barley. Okay, and that's barley that has been malted, and I'll explain what malting is a little later. This supplies the carbohydrates in the brewing process that the yeast will later consume, and they'll produce the carbonation in the beer, the alcohol. The grains also add, there's a flavor that's called malty, and there's some sweetness from the malt, because not every single carbohydrate in beer gets completely utilized by the yeast. Okay, hops are the ingredient that bitter the beer. If you made beer without hops, it would be just a little bit on the sweet side. So bitterness complements that, provides a little bit of a balance, and it also contributes an aroma to the beer, which is usually has a floral component, but may also be piney, citrusy. There's some new hops out of New Zealand right now with sort of tropical fruit edges to them. So there's a lot of different aromas that come with hops, but generally the basic one is floral. There's the yeast, of course. Yeast, they ferment a sugary solution called wort, and the fermentation yields ethanol, carbon dioxide, and heat. And of course, ethanol and carbon dioxide are two, the buzz and the fizz in beer, important parts. And then yeast, although they're an important ingredient in beer, they generally aren't present to any extent in the final product. And the final ingredient is water. That's the most abundant, but there's also sort of the least to say about it. It's a solvent. It dissolves everything. Everything in the brewing process happens in an aqueous solution. Okay, the 4 steps to brewing are malting, mashing, boiling, and fermenting. Malting is something that's done generally outside the brewery these days. Brewers used to malt their own grains, but now they don't. And I'm going to explain it in a little more detail later, but for now, let's just say the malting prepares the grains to be used for brewing. In mashing, crushed malt is mixed with hot water. In this process, the starch from the grains gets dissolved, and then enzymes from the malt begin degrading that starch. Okay, and they break it down into simpler sugars, the most abundant one being maltose, which is 2 glucose residues bound to each other. Okay, at the end of mashing, you're left with a sweet solution called wort, and it's blended, it's mixed in still with the spent grains, and so there needs to be a process to separate those two, and that process is called lautering, and I'll also talk about that in more detail later. Okay, so once the sweet wort has been separated from the spent grains in the mashing process, it needs to be boiled. So it's the wort goes to the kettle, which is the vessel used for boiling, and it's boiled. During the boil, the hops are added, and the compounds that make beer bitter are extracted from the hops at that point. It also boils volatilizes the essential oils in the hops, which give it its aroma, and a lot of other things also happen in the boil. With one of the most important being the wort is sanitized. Later on in the fermentation process, brewers want brewer's yeast, Saccharomyces Cerevisiae, to be the only organism alive in their fermenting beer, and so the boil kills off anything that's going to live in wort or beer is killed by the boil. Okay, the last of the 4 steps in brewing is fermenting. Okay, after the boil, the wort is cooled, and yeast is added to it. The yeast then consume the sugars, which are again primarily maltose, with smaller amounts of maltotriose, which is a 3 glucose residue saccharide. There's trace amounts of glucose and fructose, and there's also some larger carbohydrates that the yeast can't handle. But the yeast, of the ones they can handle, they ferment them, and brewer's yeast are homofermentative in that they only follow one fermentation pathway, and that's the one that leads to ethanol and CO2. They don't produce lactate. Okay, immediately after fermentation, the product you have is called green beer. Green doesn't refer to the color; it just means like unmatured. So depending on the type of beer, some beers need to be matured a little while to get rid of their green flavors. This is especially true with lager beers. Many ales, however, at the end of fermentation may need just maybe a day of conditioning, or some might be just ready to go. Okay, so that's the very basics of the 4 ingredients and the 4 steps. So now let's go back and look at those in some more depth. Okay, the most commonly used grains in brewing are barley, maize, rice, and wheat. Okay, barley is used in almost every beer, with only a couple exceptions. A beer is either all barley or mostly barley with some other. Maize and rice are used as unmalted ingredients in what might be called American Pilsners, the pale, very fizzy beers that are typified by the biggest breweries. Wheat is a very common brewing ingredient, usually used in smaller amounts than barley. And rye, oats, and sorghum are also used in some beers. In Africa, actually, they brew a lot of like all sorghum beers, but they're less common in the Western world. Okay, barley is a grass, and more specifically, it's a cereal grain. In the Northern Hemisphere, it's grown, well, in both hemispheres, it's grown in cooler climates. In North America, it's grown mostly in the northern United States and a little bit in southern Canada. Barley, when it's harvested, it needs to be dried, and then it's stored in grain elevators, and it's malted year-round. This differs a little bit from wine production in that wine grapes are harvested and they're made into wine ideally like before noon that day, and that happens with the wine harvest happens once a year in each hemisphere, northern and southern. But there's 2 grain harvests every year, or 2 barley harvests every year for winter and spring barley, and barley is stored in elevators, and malting occurs year round. So there's less of a seasonality to brewing than in wine. Okay, in order to be useful in brewing, grains need to be malted. Okay, malting transforms the seed, usually barley seed, into barley malt, and it includes these steps. There's initially some steps of steeping and germination. The barley is kept in vessels where it's underwater, then the water's drained out, then they fill it again, then they drain it, and this is the steeping phase. And eventually the malt, which contains very little moisture to start with, picks up enough, or the seed, I'm sorry, picks up enough moisture to sprout. Okay, when it sprouts, on one end, the little rootlets come out, the seed, and the other end, the coleoptile comes out, which is the what's going to be the stem. Right at that stage, there's actually a certain distance of the coleoptile that they measure to determine. Then they stop the process. They remove the rootlets and coleoptile and dry the malt down, and then they kiln it. Okay, kilning is basically heating. You can think of it as very lightly toasting the malt. Okay, and this develops some Maillard products. If you're cooking, those are on the outside of the grain, and the sum of the effects of malting are that the grain is softened up. Like if you were to try to chew barley seed, you'd be in danger of breaking your tooth because they're very hard. But you can chew a kernel of barley malt. It's just fine. Also, the kilning adds a nice flavor. If you were to steep a bunch of just barley seed in water and taste it, it would just taste sort of grassy and not particularly good. But if you steep a little bit of barley malt in water and taste it, it has a pleasant malty taste. Okay, the other thing malting does is it develops the enzymes that are going to be used later in brewing. There are 3 main enzymes, and we'll get to that, that come from the malt, and these are not present in any great degree in barley seed. But once it's starting to grow, then the barley needs to start breaking down its, the barley thinks it's in the field and needs to start breaking down its, starch to grow, but so it produces enzymes, and then what happens is the grain gets dried, which preserves the enzymes, and then it gets kilned, which degrades them somewhat, but not enough that there's still enough left over for brewing. Okay, typical recipe for a beer, a grain beer, or grain bill. If you're thinking of like an American Pilsner, fizzy yellow water beers, that's generally 70% malted barley and 30% unmalted corn, rice, or both. In North America, the type of malt used for that is 6-row barley malt. This is a type of malt that has a higher protein content and therefore a higher enzyme content. And the higher enzyme content is needed because the unmalted corn and rice don't supply any enzymes to the mash. So North American brewers use barley with more enzymes, which they call higher diastatic power. Okay, in contrast, a typical American craft ale or any beer brewed in Europe is going to basically be 100% malted barley, and that'll be with 2-row barley. So 2-row and 6-row being two different arrangements of seeds along the rachis, which is the central stem of the seed head. Okay, so hops, the next ingredient on the list. Okay, those are a vining plant in the order Rosales. The genus hops belong to Humulus is actually the most closely related genus to cannabis. Cannabis, of course, has hemp and marijuana and the cackberries too. Can't remember, but that's what hops are related to. Although they're related to cannabis, they don't contain any THC. There are 3 species of hops in the genus Humulus. Of those, only Humulus lupulus is the one that's used in brewing, or at least the one that's used in brewing to any extent. People have tried experimental beers, the others, but hops are grown in fields called hop yards, and these fields they're enormous. They stretch on forever, and there's trellises there that are about 9 meters tall, about 30 feet, and the hop vines climb up those to the very top, and then they're harvested once a year by machines. The machines come by and just cut down the vines and everything, and then they're brought into a processing plant where the cones, which are the actual agricultural product, are separated from the hops via some machinery. Separated hop cones are then dried. They're dried in these giant ovens. They're building-sized ovens called oasts, and then they're put into bales. So whole hops, whole cone hops are used in some breweries. Other breweries use pellets. Pellets are another product where hops are basically crushed, mixed with a binder, and extruded through a die. And they make these little pellets, which are very they have better storage capability than whole hops. So there's some brewers like that. And a lot of hops are also turned into hop extract. Some of the biggest breweries don't use hop cones or hop pellets, but they used an extract of alpha acids that have been extracted via CO2, and this they use in their beers because the stability of hop extract is extraordinary. It stays for years, and also it lacks any varietal character. You can supply just a completely blank bitterness to beer with them. An interesting thing about hops is that they're the least efficient agricultural product on the planet. If you weigh the weight of the cones from a hop vine and then weigh the vine itself, that's a tiny number. The first divided by the second. You have to grow a lot of leaves to end up with enough cones to be worthwhile. Kind of interesting. Okay, the important compounds that are in the hops are alpha acids and the various essential oils. Okay, and both of these are found in glands called lupulin glands. Okay, if you look at a hop cone and tear back the little bracts, which are the tiny quote unquote leaves in the cone, you'll see like little yellow dusty spots. Those are the lupulin glands, and they contain both the alpha acids, also beta acids, which are a related compound, and the oils. Okay, another sort of gee whiz thing about hops is that if you ever go to a hop farm, they're enormous for one thing. Hops growing up and down rows, stretching as far as the eye can see. But if you're in a block of a single variety of hops, like the Cascades, all those hops you see are a single plant. Okay, because hops are propagated commercially via rhizome. Rhizome is like an underground storage organ for a plant. And what happens when a plant is bred and developed for brewing, and they decide, yeah, that we're going to make this variety available, it's propagated by digging up the rhizomes every year, every other year, breaking them into 2 or more pieces and planting them and growing them. So and so every field you see it's entirely female hops, because the males don't produce cones that have any brewing value. And so if you go to a hop field, it's all one plant, all female plants, or well, all female one plant. And since brewers don't or hop growers, they'll get rid of any male plants in the area, because if the hop cones are fertilized, their brewing value goes down. So a sort of weird thing about a hop yard is it's one giant female plant that's never going to reproduce, and there's never going to be another plant like that either. Because, like, for example, Cascade, if you were to take the 2 parents and breed them that made Cascade, it wouldn't make a Cascade hop. So it's sort of like every hop variety, it's interesting, is singular, and it's never going to reproduce. It's interesting though. This is beyond the scope of things, but it's cool to me. There's clonal evolution in clonally propagated hops, but we don't have time for that. Okay, after malt and yeast, the next ingredient in beer is, or after malt and hops, sorry, the next ingredient is yeast. Okay, Saccharomyces Cerevisiae, or brewer's yeast, is single-celled ascomycete fungus. Okay, it's a facultative aerobe. In the presence of oxygen, it'll respire aerobically. In the presence of, or in the absence of oxygen, it ferments. And in beer, of course, it's used for its ability to ferment sugars. And although yeast is an ingredient of beer, there's very little there's very little yeast in the final beer. Most major breweries filter it out completely. Some breweries leave a small amount of yeast, or they actually re-add some for bottle conditioning or can conditioning or keg conditioning. But generally, even then, there's not much yeast in the final product. Okay, finally, water. Water is the most abundant ingredient in beer. For quote unquote ordinary beer, around 5%. That means 95% of the beer is water. Water is a polar solvent. Living things depend on water, so water dissolves almost everything in living things. So therefore, it's used in brewing to dissolve all the parts of barley and all that. To make 1 volume of beer, it takes about 7 to 8 volumes of water. So for every liter of beer, it takes 7 to 8 liters of water to make. And most of that excess water, the stuff that doesn't end up in the actual beer, goes towards cleaning and sanitation. A lot of breweries right now are starting to assess their ecological impact, and they're looking for ways to lower the amount of water that they use. It's also beer is 95% water and it's shipped all over. So shipping water places, especially when there are droughts in many places the breweries existed, it isn't as maybe as ecologically sound as it could. So brewers are also looking at ways to dehydrate beer and have it reconstituted, on site. But those experiments have not gone very far. So yeah. Okay, so that's a little bit closer look at the 4 ingredients. Let's go to the process of brewing now. Got about 15 minutes left before questions, so I'll go pretty quickly through this. Okay, malting, since it's not done in breweries, I'll leave the previous description as good, but anyone can ask me questions later. That brings us to mashing, which is the first step in brewing that's actually done in a brewery. Okay, and before the grains can be mashed, they need to be milled. Okay, and this can be done on a variety of different kind of mills. And generally in the smallest breweries, including home breweries and brew pubs, malts are milled on a 2-roller mill. And basically a 2-roller mill just has 2 rollers. You can think of it sort of like a rolling pin, and they're set right next to each other. So there's a small gap in the middle. The gap is smaller than the diameter of barley kernel. So when malt is put through it, it crushes it. It splits it open. It crushes the endosperm, which is the starch-containing part of the grain inside, and it also breaks the husk up. Okay, a 2-roller mill, basically one of the rollers is powered, and that just pulls everything through. 4-roller mills are kind of interesting. The grain initially goes through the first set of rollers, and then it falls on a vibrating screen. The smallest particles fall through to the grist case. That's just the part of the mill that holds the crushed grain. But the larger pieces sort of get vibrated down and fed through a second set of rollers, and so you get a more uniform crush. And then the absolute largest breweries use 6-roller mills, which are crazy contraptions. You start out with a 4-roller mill idea, but the medium-sized pieces from both the first set and the second set of rollers get fed into a third set of rollers, and the quality of the crush from a 6-roller mill is higher than the ones at the lower, just because it produces a more equal particle size, and that has some benefits in brewing. It's interesting. There's a major trade-off in milling, especially if you do dry milling, and that's that the more finely you mill the malt, the starch gets broken up, and the more quickly and completely the enzymes can go to work, on the dissolved starch. The larger pieces takes longer for them to dissolve, and it takes longer for the enzymes to work. On the other hand, that would lead you to think, well, brewers should just crush the grain into flour. But when it comes time to separate the wort from the spent grains, the larger the husk pieces are, the easier it is to drain the lauter time. And here's the thing: if brewers had all the time in the world, they would just settle for having a longer time, collecting the wort. But most breweries, big breweries, run 24 hours a day, 7 days a week. There's 3 or 4 shifts, and they're just cranking through brews. And so anything that slows down the process is not, acceptable. So in those breweries, if they dry mill, there's a compromise between tightness of the mill gap that to get the starch particles as their smallest, but yet not break the husk up so much. One sort of solution to this is called conditioned malting, And in conditioned malting, when the malt falls into the first set of rollers, it goes through a stream, a stream of steam. This heats the outside of the husk just a little bit, and they absorb just enough water to become leathery. So when it goes to the mill, instead of shattering the husk into a million pieces, it sort of squishes the starch out of the husk, which stays in like one or two pieces, and this yields you can close the mill gap, and you can end up with smaller starch particles but still larger husk pieces. And so a lot of breweries, if they can afford to install that in their brewery, do that. Okay, so once the malt is crushed, it's going to be mixed with hot water, but that hot water needs to be treated, and treated brewing water is called brewing liquor. One of the biggest things that needs to be done is the water needs to be treated to remove chlorine compounds. In municipal water plants, chlorine is the primary disinfectant used in the plant, and monochloramine is the secondary disinfectant that's used. It's added to the water before it goes into the pipes. Monochloramine is still a chlorine compound, so it has some disinfecting capabilities, but it doesn't. It's not as volatile. It doesn't evaporate as easy as chlorine, which is good for water treatment people, but bad for brewers. So the chlorine needs to be removed from the water, and that's usually done by carbon filtration, although it can also be removed chemically. Also, for reasons that we'll get to, brewing liquor is better if it has enough calcium ions in it. Again, we'll get to the reasons exactly, but it benefits it to have a certain amount of calcium in it. Some waters naturally have that, but if not, the brewer needs to add a little bit of calcium to his brewing liquor, and generally that's done by adding either calcium chloride or calcium sulfate. Add these to the water, they dissociate, and the calcium ions that are required in brewing are there. For most brewing liquors, between 50 and 100 parts per million calcium is recommended. Okay, something you don't, or a type of ion you don't want in brewing liquor to any extent, or to any large extent at least, is carbonates. Carbonates in beer prevent it from, during the mash, prevent it from lowering to the pH that it needs to get to. And so if there's significant carbonate content in your water, for example, if you've got a water from a limestone reservoir, that needs to be eliminated, and that can be done by reverse osmosis, or it can be neutralized by acid. For pale beers, the bicarbonate content, bicarbonate's the form of carbonate that's most present at the pH of the mash. The bicarbonate content should be under 50 parts per million. For darker beers, it can be higher, perhaps 100 to 150 parts per million. But for pale beers, it should be pretty low. Okay, so once you've got the grains milled and the water treated, they're mixed together, and they're generally held at a temperature of 64 to 72 degrees centigrade. This would be what's called a single infusion mash. In this mash, the grains are held at a single temperature, a lot of times for up to 60 minutes, and that dissolves the starch, gives the enzymes time to degrade it, and so that's the most common form of mashing these days, and it's in fact the type of mashing that maltsters produce their malt to be mashed at. There's other types of mashing, and they're interesting, but I've only got 10 minutes left. Okay. So in the mash, the starch is dissolved, and barley starch is a polymer of gluten.
Apologies, folks. It appears we may have connection.
With
Second.
hello, can you hear me?
Yes, we can now.
Okay, was I think I was gone for a minute.
Yeah, just a second.
Oh, okay. So anyway, I was just talking about barley starch. There's two kinds, amylose and amylopectin. Amylose is a straight chain of glucose residues. Amylopectin is, a central straight chain of glucose molecules with other amylose chains hanging off it. Okay, and there's three enzymes that do most of the work on the starch, at least in the mash. Okay, and these are beta amylase, alpha amylase, and an enzyme called limit dextrinase. Okay, beta amylase attaches onto the reducing end of an amylose strain. Okay, there's one end of an amylose strain that's capable of donating electrons to a reaction, and that's called the reducing end. It's the end of the glucose is a reducing sugar and has a reducing end and a non-reducing end, and so the amylose chain and the last glucose has on one end has a reducing end, and so beta amylase attaches to the reducing end of the starch, and it cuts the second bond in it, so it releases two residues of glucose linked to each other, and that's the disaccharide maltose. Alpha amylase binds to starch at random, and if anywhere along an open stretch, it won't bind right at a branch point in amylopectin, but in any open stretch of glucose residues in an amylose or amylopectin molecule, alpha amylase will go and it'll break the bonds there and produce two, in an amylose chain, it'll produce two amylose chains. And in brewing it's the idea of one of the main benefits of alpha amylase is every time it acts, it produces, it takes a molecule that had one reducing end, and now you've got two molecules and there's two reducing ends. So the more alpha amylase acts, the more opportunity beta amylase has to act. And then the final enzyme that's not really super well characterized is in brewing, they call it limit dextrinase. And this is one that just detaches the side chains of amylose from an amylopectin molecule. Okay, the bonds in starch that are broken by amylase enzymes, they're broken by a process called hydrolysis, which translates to water breaking. Okay, what happens is the molecule brings, or the enzyme brings a molecule of water and positions it right next to the bond that it's going to break, and a hydrogen ion goes to one of the glucose residues, and a hydroxyl group goes to the other, and so forth. The water is split into the H plus and the OH minus, and then those bind with the sugar, and then the bond between those two sugars is no longer present. This leads to the kind of interesting result that if you take the dry weight of the sugars produced by hydrolysis, it's actually greater than the weight of the starch that they're produced from, and that's because the weight of the water molecules, gets counted in with the weight of the sugars, and so it's therefore technically possible for a brewer to get over 100% extract efficiency, to get more sugars out of the grain than the starch included. Although in practice that doesn't occur, though, that you can do it in a test mash where you grind the grain into just nothing and mash for a day. Okay, commercially, the mash is stirred continually. It's in a large circular vessel, and there's just rakes that move around and around. That helps the mash produce or to proceed more quickly just by agitation. And in the first several minutes, the pH of the mash drops generally down to somewhere between 5.2 and 5.8. And this is as a result of calcium ions react with phosphate ions in the mash. And what happens is this releases hydrogen ions, or really hydronium ions, but hydrogen ions and calcium phosphate, which is an insoluble product that just drops out of solution. So that acidifies the mash, pH drops, and that has many or a variety of positive benefits for the beer. However, the drop in pH is resisted somewhat by the presence of amino acids in the mash. The amino acids each have a buffering point, a pH point where they buffer the solution at, and most of those fall between mash pH and boil pH. So as the pH drops, it's retarded a little bit by the presence of amino acids, but there's still enough acidity produced by the reactions between calcium phosphate to drive the pH in the mash down to about somewhere between 5.2 and 5.8. Okay, so after the mash is completed and the starch is degraded to the point that the brewer wants, it's time to separate the grains from the separate the spent grains from the wort. Okay, and this is usually done in a lauter vessel, lauter, L-A-U-T-E-R. And before it's done, the wort is recirculated. Wort is taken from the bottom of the vessel. A lauter tun generally has a false bottom, which is like a sieve, essentially. It lets liquid flow through, but it retains the grain solids. So wort is drawn off from the bottom, pumped to the top, and it recirculates, and that just filters out any larger grain solids from the wort and clarifies it visually. I mean, looking at it, you can see there's an interesting, and I see I'm coming close on time. There's an interesting trade-off in lautering that after the wort is run off, the brewers rinse the grain with water, hot water, which is a process called sparging. And of course, the more sparge water you add, the more sugars you rinse out of the grain bed. But conversely, the more energy it's going to take you and to boil that wort down to remove all that water. So there's a break-even point there. And with fluctuating grain costs and fluctuating energy costs, the big brewers, have a set of equations that they solve that tells them where's the cheapest point along that line. At what point does getting more sugar from the grain just mean we're paying more energy to boil it away? And so they can calculate where that is, but that also has to fall within a range where they can still make the kind of beer they want to make. Okay, so let me really quickly wrap up the rest of what I got here. So then there's boiling. Boiling sanitizes the wort. It lowers the pH. It forms these products called hot break, which are either coagulated proteins or protein tannin complexes. It extracts the bitterness from the hops. It volatilizes the hop oils, and it also reduces the wort volume. Okay, after boiling, the wort needs to be cooled because you're going to add yeast to it. Yeast are living organisms, and you can't throw them in near boiling sugar solution and have them survive. So the yeast is chilled, usually by a plate chiller, which is cool, but I don't have time to explain it. So that leads you to fermentation. Okay, fermentation. The chilled wort is aerated. Oxygen is added to it. Okay, this is because in order to grow, yeast cells need molecules called sterols in their cell wall to promote flexibility. And aerating the wort gives them that oxygen so that they can build healthy cell walls and reproduce quickly. It's one interesting thing about adding oxygen is that although yeast is a facultative aerobe, adding oxygen to beer wort doesn't make it switch over to aerobic respiration. Something called the Crabtree Effect is the observation that in high sugar solutions, yeast still prefer to ferment rather than switch to aerobic respiration. And the idea they think behind that is that the yeast, when they produce ethanol, ethanol is a poison. Above about 0.5% ABV, most organisms can't live in that. So they think the organism or the yeast prefer to ferment because then they kill off all their competitors. And although they're using the yeast for fermentation and they get far less, ATP per molecule of glucose, they, completely monopolize their source. Okay, so beer is fermented, then it's conditioned, and there's a lot of details on the handout about fermentation and conditioning. Finally, it needs to be carbonated. Okay, when beer is done fermenting, it's saturated with CO2 at the pressure, at atmospheric pressure, but more CO2 needs to be added to it, and then it needs to be put in a pressure vessel so that it's actually fizzy. And there's various ways this can be done, and brewers are seeking more ways to, because a lot of times, or in older breweries, they tons of CO2 comes off during fermentation, and then they would buy CO2 tanks to add it back to the beer. And but for environmental reasons and others, they're working on capturing CO2, cleaning it, and using it to carbonate beer. And finally, beer is a food product, so you should store it cold. Refrigerator temperature is best. If you do that, it should have a shelf life of about 6 to 8 months, depending on the beer, and some estimates vary because some people are more sensitive to staling products. But that's a basic overview of brewing. I've got a book coming out, as Matt mentioned, called The Science and Craft of Brewing, or Science and the Craft of Brewing. It should be out next year, fingers crossed. And that has everything I've said, everything in this handout, plus a lot more. So at this point, I'd welcome questions.
All right, so we have a few here. One is, oh, go ahead.
Oh, should I scroll through these questions, or do you want to read them?
I can read them to you.
Okay.
So one is, do IPAs generally use more hops than average? Some of a skunky smell. What is that caused by?
IPAs use much more than average. That's their whole thing. They're the skunky smell. That's actually interesting. It's there's a reaction that hops have when light strikes them that causes a molecule to be released, and that molecule is actually, it's either the one or very similar to the one that's released by actual skunks. And an upshot of this is that the particular wavelengths of light that produce that skunky smell are blocked by brown glass. That's why most craft beers you see are in brown bottles as opposed to green bottles. Green bottles let that those wavelengths through, which is why if you've ever had a Heineken in a green bottle, it probably had sort of a skunky aspect to it. But yeah, IPAs use more hops, and then if they have a skunky flavor, that's because they're what's called light struck. It just some UV rays have hit them.
Gotcha. And you kind of answered this next question. Does the chemistry of water greatly affect the brewing process?
Yeah.
We may have lost Chris for a second here.
Oh,
sorry, could you repeat that, Chris?
Okay. Important thing, arguably, but probably is getting rid of any chlorine products in your water. The second is getting enough calcium in your water. Magnesium also has some of the properties calcium has. And limiting the amount of dark beer, pale beers. And yeah, these can, very definitely, you can taste it if you're a home brewer and you go from like not bothering to treat your water to treating it and making the mineral content right, an impact on the flavor and aroma.
All right. Here's what question says, Texas beer called More Cowbell, which is very interestingly beer because it has an IBU of 118. And it says, is there a bell curve of hoppiness or bitterness flavor associated with IBU?
Yeah, IBUs are international bittering units. They're a way that brewers specify how bitter a beer is. One of IBUs is actually they determine it spectrophotometrically, but a lot of brewers just calculate it from some sort of calculations. I would very highly doubt that beer is actually 118. Although the fact that it doesn't have hop flavor lends me, I mean, it could be if they used hopped extract. That would make a very bitter beer, but it wouldn't have a lot of hop flavor, or wouldn't have any hop flavor or aroma. I'll have to look into that. That's interesting. I've never heard of that beer.
All right, and I'll just mention, if you have questions that we can't get to, Chris did mention he will follow up and send me the answers to those questions. So we will document it all and try to get those answers back to you. So the next question we have here is fairly experienced extract home brewer. Many of my brews come out with a bit of a Band-Aid flavor. It tends to mellow the bottle with bottle conditioning. Any idea how to reduce this?
Yeah, Band-Aid flavors are typically they're from phenolic compounds, compounds based on chemicals that are related to phenol. That's usually due to contamination. The way to reduce this would be to pay extra special attention to cleaning your brewing equipment and sanitizing everything that comes in contact with wort or beer.
All right, thanks. If you have the very basic equipment, what's the best piece of equipment to buy or upgrade to notice an improvement in your homebrew beer?
That's a good question. That's a very good question. I would say in terms of improving the quality of your beer, getting a chest freezer or refrigerator with an external thermostat so you can hold your fermentation temperatures. That's going to be the best way to improve your quality. The best thing to get if you want to just make your life easier is a basic kegging setup, because it's a lot easier to bottle your beer in one keg than in 2 cases of 12-ounce bottles.
All right. What's the basic difference between making ales and lagers?
Ale yeast and lager yeast are 2 different species, and ale yeast works best at a higher temperature than lagers. Lagers also require a conditioning period afterwards, whereas ales may or may not. So it's essentially a temperature difference or a temperature difference that the different yeasts work at, and how quickly the beer is produced. Back when the sort of brewing or craft brewing revolution started out, one of the big selling points was that brewers could make an ale, and they called it grain to glass, in 7 days. You could brew an ale, and 7 days from your brewing point, you were serving it in your brew pub, whereas a lager takes maybe 7 to 10 days to ferment, and then like a minimal of 21 days conditioning time.
How do you know when the fermentation process is completed?
If you're a home brewer, you'll be fermenting like either in a bucket or a carboy, and you'll have an airlock on top, which lets gases out, the CO2 that's being produced, and it sort of bubbles through a little tube. And what you'll see is that the bubbling in that will slow down and slow down and slow down. And then you can also look at the specific gravity of the beer. You can take a hydrometer, which is a little instrument that just floats in
folks. If he comes back here.
You'll know. Did you get that?
Uh,
Or was I gone?
Hey, Chris, can you hear me? All right. A few more questions
I can
here.
hear you.
What's the difference between a hazy beer and just a beer that's been unfiltered?
Oh, God. Yeah, I don't know. A lot of sort of grumpy old brewers like me hate those hazy beers. And it's actually not just simply, although a lot of people like to make fun of it, it's just beer that the brewer didn't have enough skill to make not hazy. But it's actually they add starches to those beers to make them hazy, and they've got various compounds and starches that actually have haze that hangs in solution, because a lot of haze is especially in a cold solution is just going to drop out. There's a whole set of ingredients that they add to hazy beers that are generally starch based to make them more hazy.
Okay. And then how does the brewing process differ for producing a sour beer?
Oh, that's a good question. Yeah, sour beers are a favorite of mine. The process is very similar, or can be, but it differs. There's actually a couple ways you can do it, but the way most of them are done is you go through and ferment the beer normally, and then you add some lactic acid producing bacteria to it. Lactobacillus, there's a couple species of lactobacillus that are used in brewing sour beers. There's a couple, or at least one species of pediococcus, and these work on the residual carbohydrates left behind by the yeast, and slowly sour it. Okay, like if you brew a beer, well, like a traditional Belgian sour beer is a lambic, and so those ferment like within a week, but then they pick up lactic acid bacteria from the barrels they're stored in, and that sours. And generally, they leave them to sour for a year, and then they'll either add fruit to them or package them as pale beers. And yeah, there's more to it to the lambics than that, but basically, yeah, you let bacteria sour the beer after the normal fermentation has been done, and that souring takes quite a while.
Awesome. And then I think the last question, can you provide the differences between, I guess, West Coast IPA, New England IPA, and maybe some other IPAs? What sort of defines the differences between those?
I don't know. I could. There's a homebrewing group called the BJCP who deal with all that kind of dull stuff of breaking beers up into different categories. And I mean, basically, a West Coast IPA it's a relatively clear but relatively hoppy IPA. It's more hoppy than an old school English IPA. New England IPA is the hazy version, and I don't know. There are people who like to split things up into a million categories, and that of the parts of brewing, that's what bores me the most.
So I guess what might be neat is, do you have any informational links you would recommend for home brewers? Maybe that's something that you can send to me and I can send out in a follow-up email.
Yeah, I mean, there's a lot of good resources for homebrewers. I would say John Palmer wrote a book called How to Brew, amazingly straightforward title, and that's a good one. I have a website called Beer and Wine Journal that has a lot of homebrewing content on it. There are just oodles of brewing forums out there. The downside of that is that a lot of people write and answer a lot of questions, and they actually don't know anything about brewing, and so you have to sort of know like which answers to trust, which takes time. But yeah, the How to Brew book is really good. That's where I would start if you were into homebrewing.
Awesome. Well, we are at the 101 mark. So I just want to thank you, Chris, for joining us for this call. Thank you to everyone that attended. We will be This has been recorded, so we'll send out the link to it if you wanted to be able to share it with your friends. We are doing a contest, so we will be doing a drawing, and you have a chance to win one of Chris's books. So we will let you know directly if you are one of those winners. So thank you all for coming. Again, stay tuned for future talks that we'll have with the LLESA author series. But everyone else have a great afternoon, and thank you for your time.
Yeah, thank you, Chris.
Thanks, Matt.